Hello, we meet again. So in the last lecture, we talked about gene knockout technology. Now this is the first edition of targeted gene manipulation in mouse. Okay. Now this process has certain disadvantages. Like, let me elaborate it for you. And those which are from my own life's experience, so that it is easier for you to understand. Say, as I explained earlier, that you want to study a very critical signaling pathway. It doesn't matter. It can be BMP, it can be TGF-beta, it can be Wind, it can be hedgehog, any key signaling pathway you think about, which are called toolkit signaling pathways, or some critical transcription factors. Any genetic pathway which has roles in more than one theaters. Okay, maybe it is important for muscle development, maybe it is also important for kidney development, maybe it is important for so many things. Now, in that kind of a setup, if we do this kind of a knockout, then what happens is that the gene is lost in every tissue from the first two-cell stage embryo itself. From the zygote itself. Now, the gene that you were knocking out, if it is critical for very early stage embryonic development processes, like maybe without it you will not form the nervous system, maybe without it you will not form the heart, or without it you will not form the vasculature, then what will happen? Then the mouse will not survive. It will not survive beyond very early embryonic stages. So if the gene that you are trying to knock out, if it has functions later in the at later stages of life in the animal, maybe late embryonic stages, or in the newborn, or in pre-pubertal, or in adults, you will not be able to analyze any of those. Why? Because the embryo was long dead. That is one kind of a problem. The other kind of a problem is that in the same general tissue, the same gene may have different functions at different stages of development. Okay, maybe early on it is important for some patterning events, like patterning, meaning that what will be the relative positioning of different cell types. Okay, and maybe later on it has some differentiation role in differentiation. So if you want to dissect the two, this is not the tool that will take you there. Why? Because the gene is knocked out from the very beginning. So to avoid, to tackle these kind of problems, there is another method of gene knockout, which is called conditional knockout. Conditional knockout, as the name suggests, that it will be knocked out depending on the condition dictated by the researcher, you. Okay, so let me explain to you how this works. So I am giving you example of a specific case. In this case, it is a Cree-Loxp mediated knockout system. So this is called the Cree-Loxp mediated conditional knockout system, where Cree is an enzyme. Okay, it's a site-specific recombinase from bacteriophage P1. It's a bacteriophage, that's all that you need to remember. Now, this is a site-specific recombinase, right? That means that it recognizes a particular sequence, and through homologous recombination of that sequence, it recombines out the sequence, the intervening sequence between the two homologous sequences, and that's how it works. Now, this conditional knockout system is always a two-component system, meaning there will be two different genetically modified mouse strains that will be needed for effecting conditional knockout. So one mouse, which we call the deleter mouse, there is the like the LOXP mouse. There what happens is that you simply insert two LOXP sequences. LOXP is the recognition sequence for the Cre recombinase. Okay? You just insert two LOXP sequences in the intron. Why in the intron? Because if you insert in the intron, the gene, the WRF, the protein coding sequence will not be affected. So you do not expect the gene function to be affected at all. So a LOXP mouse where you have inserted two LOXP sites on either side of a critical exon, one exon or two exons, or the whole gene, doesn't matter. That mouse will be phenotypically normal, right? Genotypically, of course, LOXP has been inserted, so genotypically it's slightly different, but it will be phenotypically normal. But upon condition, that is, if the cells where this genetic architecture is there, which is every cell in the mouse, because you have done it using the ESL modification method, right? So if in that same cell you express the Cre recombinase, then what will happen? Then Cre will recombine these two LOXP sites, and the intervening sequence, this exon 2 sequence, will be chucked out. So now the gene will not have one critical exon. So that is how it is a conditional knockout. The condition is presence of Cre in the cell. So that's where the utility of the second mouse line comes in. The second mouse line is called the Cre line, or the Cre mouse. The Cre mouse is simply the Cre recombinase sequence from bacteriophage P1. It will be under the influence of cell type specific promoter enhancer, so that it is expressed only in a particular cell type. Like if you go back to the previous example, you do not want BMP to be knocked out in the heart, because if it is knocked out in the heart, you will not be able to study bone. If you want to study heart only, be my guest. You do either plain knockout or this to ensure that Cre is expressed in the bone, that is in the heart, that is fine. But if you want the BMP to be not knocked out in heart, but only in bone, what do? You will use the promoter enhancer that is well known to drive expression in bone cells or bone precursor cells, meaning cells that will eventually become bone, right? Now what will happen? Now the Cre enzyme will not be expressed in any other cell type of the mouse, but only when bone precursors are formed or born, then Cre enzyme will be expressed in those specific cells. So now in the bone precursor cells, you will have the LOXP flanked exon, and you will have the Cre recombinase enzyme, because it will be expressed only there. That is the job of the promoter enhancer. Then it will recombine these two sequences, and the intervening sequence will be floxed out. We call it floxed mouse. these mice, we call them floxed mice. Okay, and when this recombination happens, we call the sequence to be floxed out. So now this, how you deliver the Cree can vary. Okay, that it can be obtained through, just like you have in previous example, you have inserted neomycin cassette in here. You can imagine that you can insert this sequence can very well be neomycin and Cree, okay, with the homology arms intact. Okay, so now you will target the first exon, will have the Cree and neomycin cassette, the first exon. Then, as you will learn when I will talk about transcription regulation, the first nucleotide of the mRNA is the first nucleotide of the first exon. Right? So then the whatever gene it was, you knocked in, instead of, like, it is just a matter of semantics, you still knocked out the first exon, but you knocked in the Cree enzyme. Now the Cree enzyme comes under the control of the same gene X. Okay, and wherever gene X was normally being expressed, that's exactly where the Cree will be expressed. So the Cree can be a result of a knock-in, or the Cree can be a result of a transgenic mouse. Okay, transgenic mouse. I will explain what is transgenic mouse here. So whether it is a knock-in or a transgenic mouse, the limited job is to deliver the Cree enzyme in the cell type that you want. Okay, as long as you can accomplish that, you are fine. That way, you will be able to flox out the essential component of your target gene only in the cell type you want. Okay. Now, this knock-in mediated Cree generation has a problem. That if for some reason, because when you are knocking in, you are knocking out an exon, right? So for the gene, say you are knocking in the locus of gene X, then it will become heterozygous for gene X. If lowering the dosage of gene X has any phenotype on its own, then that might complicate your analysis. So though it is preferred that Cree is a knock-in Cree, because knock-in Cree will faithfully recapitulate the normal mRNA expression pattern of the gene X. Like Cree will be expressed exactly where and exactly when X would normally be expressed. But it has that problem. But to avoid that problem, you can go for another method, which is called transgenic mouse. So, I will come to transgenic mouse a little later. Now, this very process can also be used to accomplish what is called fate mapping. It's a typical developmental biology application. What is this developmental biological application? I'll give you an example. See, initially, we all started with one zygote, right? Then the zygote became 2 cell, and then it became, 4 cell. Then it became many cells to become the blastocyst. Then it will go on further Eventually, you will have endoderm and mesoderm, endoderm and ectoderm. Then there will be induction of mesoderm, and then the complexity will keep on growing. Now, if you want to know that, like say, I'll give you an example. Okay? Pardon my drawing here. So if this is your hand, there are two bones, radius and ulna, and then there are muscles here, and then there are tendons and ligaments here. Okay, tendons and ligaments, they connect muscle with the bone and bone with the bone, respectively. Tendons connect muscle with the bone, ligaments connect bone with the bone. Now, if you want to know that whether all these different tissue types, muscles, bones, tendons, ligaments, where do these tissues come from? One possibility is that they form in situ, meaning they form within the limb, within the developing limb. Or the other possibility is they form somewhere else and then move inside the limb. Okay, so how would you know that? How would you distinguish between these two possibilities? So using the logic based on earlier literature, say you narrow down between two cell populations, A and B. These are the two populations from where the muscles can come from. Okay, one population is obviously the cells in the limb at a very early stage limb before muscles were born, or muscles were seen inside the limb. Let's say that is B. Okay, maybe that is one population. The other population is A, which is not exactly in the limb but nearby. Okay? If you want to distinguish between these two possibilities, what would you do? This you will do this. That is, you will take a mouse strain where there will be the genetic construct will be something like this. It is not promoter, it is promoter enhancer. Okay, so this promoter enhancer will be specific for the cell type, whether it is A or B. It doesn't, like say you are testing whether B is the cell type which gives rise to muscle. And you know that there is this gene K which is only expressed in B cells in B. Gene K is only expressed in cells of the B. So that means that the promoter enhancer of K ensures that the RNA right after it will be transcribed in B and B alone and nowhere else. So you simply take that promoter enhancer and then have this very cool structure. This is a LOXP site, this is a LOXP site, and this is not a translation stop site. This is not a translation stop site. This is what is called a transcription termination sequence. Which is what you know that. This is called the polyadenylation sequence. Okay, so it is some 1,000 nucleotide long sequence. You put that between two LOXP sites, and then on the other hand of it, you keep a reporter. The reporter can be alkaline phosphatase, it can be GFP, it can be RFP, whatever you want. Nowadays there are some very fancy looking reporters up there. Now that is one mouse. Okay, this is mouse one. This is mouse one, and this promoter enhancer is actually the promoter enhancer of gene K. So it is, you know that this promoter enhancer can only drive expression in B cell type. Okay, and Sorry. I withdraw. There is a major mistake I made. There is a major mistake I made. This promoter enhancer is ubiquitous. Ubiquitous, meaning it can drive expression of gene in any cell type. Any cell type. Okay. Typically, like actin, tubulin, the one that is used was called ROSA 26. This is the most commonly used promoter enhancer for fit mapping. Okay. So ROSA 26 is a gene that is expressed in every cell type. So ROSA 26 promoter enhancer, therefore, has the ability to drive expression of any gene in any cell type, right? So you simply make this construct. It is a ubiquitous promoter enhancer. Then under its influence, there is a reporter gene, which can be GFP, OP, anything. And the flanking region, in between the two, there is this fluxed transcription stop cassette. Okay? Fluxed transcription stop cassette. So as a result, what will happen is that RNA pol will start the transcription right here. It will go, and then when it hits here, it will fall off. So the promoter, so the reporter cannot be transcribed as long as this sequence is there. As long as this sequence is there, right? Now you use a second mouse line, which will express, now this B cells, they have this gene K, which is only expressed in B. So you take the promoter enhancer of K, under which you express K. Now what will happen? K can be expressed only in the cells of B, only in B cells and nowhere else. Right? So only in B cells, this stop cassette will recombine and will be fluxed out. Now the reporter is under the influence of the promoter, and the transcription can actually happen because the transcription stop cassette is removed. And no matter what happens to B cells, it will continue to be expressed. Why? Because this promoter enhancer is ubiquitous in nature, right? So whether, just to use little technical term, the very early limbed cells, they are called limb mesenchymal cells. So if the limb mesenchymal cells become bone, it will still be expressed there. If the limb mesenchymal cells become tendon, it will still be expressed there. If the limb mesenchymal cells become muscle, it will still be expressed there. So no matter what happens, once the recombination happens, then the promoter will continuously drive expression of the reporter. See the advantage of this process. What is the advantage? So, very early limbed looks like this, right? Here, there is no differentiated cell type. There is no muscle, no cartilage, bone, tendon, ligament, nothing. So this is what you call B. And K is a gene that is only expressed in limb mesenchymal cells. Okay? There is a gene that I can tell you like that called PRX1. PRX1 is a gene that is only expressed in limb mesenchymal cells. Okay. Now, if you use a CRE that is called PRX1 CRE, then when you cross PRX1 CRE mouse with the reporter mouse, then the reporter will not be if this is the head, and this is the trunk, this is the tail, and this is the other limb bud, whatever. PRX1 will not be expressed anywhere other than these two B regions. And because PRX1 will be expressed there, so the Cre will be present only here. So only here, this recombination will happen, and these cells will become reporter positive, GFP positive. Okay? Now, if these limb mesenchymal cells, eventually when it will go into a hand, and muscle is there, if the muscles came from these B cells, then muscles will be GFP positive or the reporter positive. If you don't see that, then you know that the B cells did not give rise to the muscles. On the other hand, there is a structure here called somite. Okay, no need to get into the detail of it. If you use the Cre driver instead of PRX1 Cre, you use something that expresses in the somites, then the Cre will be expressed only in here, only these cells will become GFP positive or reporter positive. Now, after the limb has grown out, if you find that the muscle cells are GFP positive, then you know that the muscle cells did not originate from the limb per se, but originated from the somite and moved into the lymph field. And that's what is actually true, that the muscles that you have in your limb, they did not come from the limb per se. They came from behind your back, something called the somite. They actually move in. On the other hand, the bones of the limb, they actually form in situ. So if you mark with PRX1 Cre, you will see that the bones and tendons and ligaments, they get marked. But if you mark with somite specific Cre, bones, tendons, etc. will not be marked, only muscle will be marked. Okay. Now, so how this is done, this is what it is. It is like this is a site-specific recombination. I think I'm forgetting. So here in this gene A, if you use site-specific recombinase, okay, if you use this kind of a mouse where, and when in mouse genetics, when you use this symbol, it denotes a knock-in. So if site-specific recombinase is knocked into gene A domain, then SSR will be expressed exactly where gene A is expressed, and that's where the cells will turn reporter positive. With time, you may see they are migrating, and then they are distributing in these beautiful patterns, etc. So this is how fit mapping is done. I will show you our own experiment. So GDF5 is a gene that is expressed in very early joint cartilage. Okay. And if you cross it with a reporter gene, so R26R meaning rows are 26, okay, followed by LAGC. How is it that this is the ubiquitous? There is a typographical error here. It is promoter, tissue specific promoter enhancer. Okay, promoter enhancer. So tissue specific promoter enhancer driving the Cre line, so it will be expressed exactly where GDF5 is normally expressed. You cross it with another mouse that has LAGZ or GFP under the control of a ubiquitous promoter enhancer, but there is this transcription stop cassette. You cross the two, and then you get in these cells the the reporter comes directly under the control of this promoter enhancer. Okay, and where the cells where Cre recombinase is so there are some typos here. Okay, this will be without. I'll correct it. Where the Cre recombinase is not expressed, meaning any cell other than the early joint cartilage cells, there the Cre will not be expressed, so therefore this top cassette will not be removed, so those cells will not be marked. Okay, so this is how we can lineage trace what happens, like how the joint cartilage cells from where did they come from. Now, so that is the fundamental. Okay, now how do you make transgenic mouse? So transgenic mouse is fundamentally different from knockout or knock-in mouse, both how it is generated and also how the data should be looked at. So if you remember, knock-in or knockout mouse was generated by first modifying ES cells and then injecting the modified ES cells in a blastocyst, then take the blastocyst, putting it in a pseudo-pregnant mother, and expect that in some of the progeny from this manipulation, where the modified ES cell will contribute to sperm production, okay, so that it goes germ-like. Here it is not like that. Here also in vitro fertilization is involved, so sperm and egg will be put together in a petri dish, they will be fertilized, and then you get the first cell of the embryo, zygote. There you will have, and it is very easy to recognize, which is called the female pronucleus. Remember, there are two haploid cells, one sperm and one egg. First time when they fuse, then therefore there are two different nuclei. Okay. From second cell stage onwards, there will be one nucleus which is diploid. This first time, there are two different haploid nuclei, and of which the female nuclei looks much bigger. So it is much easier to locate. So you take a modified DNA, you take a yeah, so in these cases, what you do when you are doing this transgenic mouse, say you want to express GFP in tendon cells, okay? So you know the tendon specific promoter enhancer, okay? Tendon promoter enhancer. Then you put GFP and make a DNA construct. Of course, you do it in a DNA construct. Then you will originally have a plasmid, and then you will cut the plasmid and then put a linear DNA and inject it in the pronucleus. When you inject it in the pronucleus, it says that prior to fusion of male and female pronuclei. So when you inject it in the pronucleus, it will undergo random integration in the genome. Random integration in the genome. There is no guarantee exactly where in the genome it will go. Neither is there a guarantee how many copies of the DNA will go. Okay, so it is a little bit, like shotgun fire. It's not a sniper rifle. Your sniper rifle is your knockout or knock-in. This is like shotgun. Okay? Now, the problem that happens because of this, say this is a very large chromosome, and here the natural chromosome has a very strong promoter enhancer. Okay, and accidentally your construct got inserted right over here. Then this promoter enhancer can overcome the influence of this promoter enhancer and express it wherever this will direct it to. So you do not have complete control over the place where your reporter will be expressed. You wanted it to be expressed in the tendon. You used tendon-specific promoter enhancer, but because of the location of the integration, that instruction, this instruction is being overridden by something else. Okay? So that happens. Sometimes it can get inserted into a silent locus, no expression. So all of these vagaries are there. Therefore, what you will have to do is you will have to test few hundreds of mice that will come out of each of these. Like each injection is an independent event, right? So one zygote injected at pronuclei number 1, you put it in, you take several such, put it in one female. Okay, these are all independent events. Okay, and you put some more in another female, and so on and so forth. And then you get the mice, the babies born, and they allow them to grow at whatever stage you want to look at, dissect, and see if the reporter is being expressed exactly where you wanted it to be expressed. This is called position effect. Okay, so position effect is a big problem when it comes to transgenic mice. This you do not have in knockout or knock-in. Like knockout or knock-in is much more stringent. Transgenic is easier to do, but a lot of chance factors are there. There are a lot of artifacts that have happened. People are now discovering, and there are lots of issues. Okay, but sometimes you will have to generate your Cree line by transgenic. Say, for example, if you are trying to generate SOX9 Cree, okay, SOX9 is an haploinsufficient locus, well known. So you cannot have a SOX9 Cree as a knock-in because you cannot afford to knock out SOX9. But you can do a SOX9 Cree like transgenic, and rigorously screen and see exactly in which mouse the expression pattern is just like what it should be for SOX9. Then you are fine. So you have to do a lot of after screening to get the right kind of mouse strain. Okay, so this is transgenic. Okay, I hope now you know what is transgenic, what is knockout, knock-in. Genetically modified mouse will be in both transgenic as well as knockout, knock-in. But if someone is meaning to say knockout or knock-in but calling it a transgenic, call it out. That's wrong. Okay, these are different. Now, I taught you about how to generate loss of function in mouse using knockout, knock-in. If you are doing experiments with chick, okay, that I do, okay, chick does not afford you this kind of knockout, knock-in, okay, or even Drosophila, etc. So there are other ways of doing loss of function studies. One is to express what is called a dominant negative allele, okay, dominant negative expression. So a dominant negative one, like in this particular case, say it's a GTPase, okay. The normal GTPase, it will be normally bound by GDP. It will interact with what is called a guanine nucleotide exchange factor, or GEF, and then the GDP-GTP exchange will happen, then it will become active GTP, then it will promptly hydrolyze the GTP, and it will come back to this state. Okay? So this constantly happens. Okay? Now, if you make a version of GTPase which will be able to bind to GDP, but GEF cannot do the exchange from GDP to GTP, then what will happen? This will remain stuck with GEF, with GEF. So this is therefore a dominant negative version of GTPase. Why? Because in the same cell, if you have the normal GTPases, they will not be, because this dominant negative GTPase cannot undergo the conversion from GDP to GTP, so they will not disengage from GEF. They will just sit there. And soon the cell will have no GEF available. All are occupied by your dominant negative allele. And generally, when you are doing it experimentally, you are overexpressing it. Okay, so if you the normal copy of GTPase, which will be there in the cell, right? It's a wild type chick embryo, so it will have its normal GTPase. But say it, in a cell it has 10 copies, and this you are expressing 100 copies. Okay, so soon it will completely titrate away any available GEF. Therefore, the GTPase will not be able to act. So this is a way of doing loss of function. In our labs, we often use a strategy. Say this is a transcription factor. Every transcription factor has, most of the transcription factors have two domains. One is a DNA binding domain, and the other is a activation domain. Okay, the DNA binding domain dictates where in the chromosome it will bind to. So the sequence specificity for the transcription factor comes from the DNA binding domain. An activation domain dictates that because of this binding, there will be activation of transcription. So the gene that it is regulating will be only expressed in the specific tissue dictated by the chromosomal sequence to which it is binding. Okay. Now imagine that this domain is replaced by something called engrel. Engrel is a very well characterized transcriptional repressor. It blocks transcription. So this fusion protein will still bind to the preferred DNA sequence of the transcription factor of your interest. Okay, but now instead of promoting transcription, it will block transcription. So all the target genes of your favorite transcription factor will now no longer be expressed. So that is the way of doing loss of function. Or you can promote the activity of your specific transcription factor by putting a very strong activation domain. Generally what is put here there is a GAL4 activation domain. Okay, and this is a very, very strong activation domain. So you override every other regulation, and as long as your fusion transcription factor is recruited to the domain of interest of your, like to the domain where, to the region of the chromosome where your transcription factor of interest is generally recruited, it will activate transcription factor, transcription of every single gene that is normally downstream of it. So these are so they're like in chick, frog, sometimes in Drosophila, these techniques are used very, very routinely. Okay, and then of course you know about RNAi technology. Okay, RNAi technology, I will not get into the history right now. I think I have a couple of lectures on that, but it is simply, like you express like the first edition of it was like this, that your gene of interest in two different plasmids, in one you express it in the transcribe it in vitro transcribed. So if it is T7 promoter, then you just add T7 RNA polymerase and it will be transcribed in vitro. One plasmid will transcribe the sense version, another transcript will transcribe the antisense version, and then you, because you are producing them together, they will form double-stranded RNA, and that double-stranded RNA you can inject, and the cellular machinery, like dicer and all of that, you have heard of it, will produce the active agent that can silence the gene. Okay? It acts both by degrading the target RNA or by blocking translation. Okay, and there are some subtle differences, like siRNA, shRNA, there are subtle differences. So that's one way. The other way is that in a DNA construct, you have two regions of sequence that are like, this is identical to your RNA of interest. Okay, so this is the sense sequence. This is the antisense part for the same, and there is something that has nothing to do with this. So that RNA will fold. This is the sense part. This is the antisense part, and the middle part had nothing to do with it. It's called a hairpin. So this hairpin will be produced in vivo, and then the cellular machinery will make siRNA out of it and will knock down. In our laboratory, we have used this technology multiple times in chick. In fact, we were only the second group in the world to use RNAi technology to knock down genes in vivo, in chick. Others have done it in other organisms. In chick, I think we were only the second group in the world to have used it, chick embryo. Okay, and since then we have done it many, many times. There are lots of complexities in it, but the basic principle is this. And now, of course, you should know that CRISPR-Cas is the new hero in the scene. Just yesterday or day before, the Nobel Prize was awarded for this to Jennifer Doudna and Carpentier, and so that is now a very, very effective technology for gene silencing, and it works at the DNA level, of course. Okay, this works at the mRNA level or the translation level. That acts at the DNA level itself. Now, I am trying to organize one special lecture for you on CRISPR-Cas. Honestly speaking, I am very old to learn new things. I have a very, very smart undergraduate student who is doing a theoretical project with me where she is learning the fundamental process of CRISPR-Cas technology and its utilization in biotechnology. And she is doing that with me. So I have requested her to prepare one lecture for you somewhere towards the end of October, where she will deliver a special lecture, not like this, to talk about CRISPR-Cas system. Okay. So there are many different spontaneous genetic disorders. They are linked with changes in gene sequence. They all have particular inheritance patterns. These are very simple. Now that you know is genetics, it is no big deal for you. You can look at it. The single nucleotide polymorphism, if you have the ability to sequence many genomes, you can use that to trace inheritance of genes and correlate it with disease phenotype to get association of chromosomal loci with certain diseases, and you can map it into the human genome. There are many things. So that's pretty much it. I do not have anything else to talk about in this context. So with this, the series of topics on techniques in biotechnology or molecular biology we have covered. Now, with this, you should be able to read any relevant scientific literature, textbooks, other materials on the web. I want you to become very familiar to understand which technology is used when, what is the outcome, how to interpret the data, what can happen, what are the ways you can misinterpret the data. All these things I really would encourage you to pick up. And once you have done all of that, it will be very easy for you to follow the next topics, where I will be talking about fundamental processes of molecular biology, like transcription, translation, regulations thereof, splicing, all of these. Because I emphasize on the history, that how we came to this point where we know what we know, and so to learn the history well, you need to know all sorts of technologies that were used in the 1980s and the 90s and then 2000 and now. Unless you have a lot of comfort level with all these different kinds of technologies, you will not be able to appreciate the scientific literature from its origin. And until and unless you know how the original discoveries were made, you will not be able to not only not appreciate the current literature, you will also, like a new scientific hypothesis is generated often by reinterpreting old data, like reading old paper, and then suddenly something strikes odd, and then you form a new hypothesis. Like the striking odd part is depends on your ability to interpret the old data. As I said earlier, and I'm saying again, that data is always right. It is the interpretation of the data that is sometimes subjective. So until or unless you know exactly how these experimental techniques work, you will not know what can possibly be artifactual, what could have possibly gone wrong. So, without knowing that, you will not be able to reinterpret an old data. So this is the reason why you should have a very comprehensive understanding of molecular biology techniques that have been used since the 1970s. Okay. So with that, I will end this series of lectures. I hope you learned and you enjoyed this. Thank you very much.